Okay, so in this lecture, we are going to discuss our icosanoids. And so these are some of the objectives, is just to understand what icosanoids are, what they do, the difference between those that are going to be made from omega-3 fatty acids and omega-6 fatty acids, what their structures are, categories, functions, etc., the different enzymes that are going to form different icosanoids, lipoxygenase, cyclooxygenase, etc. And so just as a general overview, our icosanoids are going to have certain names. We're going to have our prostaglandins, which is our PG series. And prostaglandins were first identified in the prostate gland, and that's where they got their name prostaglandin. And then we have our thromboxins, our TX series. These were originally identified in platelets. And then we have our leukotrienes, or LTs. And these were identified in leukocytes. So prostaglandins were identified first in the prostate, thromboxins in platelets, and leukotrienes were identified in our white blood cells, or leukocytes. Now we also have lipoxins. And those are LX. And these are going to be synthesized through an enzyme called lipoxygenase, which we'll talk about. And these are going to be important with modulating the other icosanoid responses. So we also have our prostanoids. Our prostanoids are specifically our prostaglandins and our thromboxins. And we have various nomenclatures associated with our prostanoids. They're going to have a number, a subscript number, which is going to be reference to the number of carbon double bonds. So we are going to have a subscript number. And then our biologically active lipids are typically going to be series 2, but not always. Okay, so they have 2 double bonds. So we also have series 1, and you'll see some with 4 as well. So here are some clinically relevant. We have prostaglandin E2. And this is known as dinoprostin. We'll have a slide about dinoprostin and its functions, but this is going to be a mediator of inflammation, and it can have both pro and anti-inflammatory. And then we have our thromboxin A2, and this is going to be stimulating platelets, and it's going to activate platelets and help facilitate platelet aggregation. And then we have our leukotriene B4, and this is going to be involved in inflammation and is thought to promote insulin resistance. So we also have a lipoxin A4, and this is going to be anti-inflammatory. And for this guy, aspirin is actually going to trigger its production. And we have a lipoxin B4, which is going to, whenever you measure levels, increased levels of lipoxygen B4, this is a signal that the inflammatory response is resolving. Okay, so the precursors of your different icosanoids are going to be arachidonic acid, and arachidonic acid is going to be made from an omega-6 fatty acid. We also have dihomogamma-linoleic acid, D-gala, and this is also from an omega-6. We have eicosapentaenoic acid, or EPA, and that comes from an omega-3 fatty acid. So arachidonic acid is going to lead to different icosanoids depending on which enzyme is going to work on arachidonic acid. So COX is cyclogen oxygenase. And LOX is lipoxygenase. And so depending if you're going to have cyclic oxygenase or lipoxygenase, you get either series 2 prostaglandins or series 4 lipoxygen. And these are going to be both high inflammatory. So arachidonic acid is typically our pro-inflammatory icosanoids, come from that precursor. Our EPA from our omega-3, again, the same two different enzymes, and you get instead a series 3 prostaglandin or series 5 leukotrienes. So leukotrienes are LT, low inflammatory, and then our D-gala, series 1 prostaglandins and series 1 leukotrienes, and these are going to be more anti-inflammatory. Okay, so we have our omega-3 essential fatty acid, alpha-linolenic acid. It is an 18 carbon with 3 double bonds. Now the endoplasmic reticulum will take this fatty acid and through a very slow process will desaturate it. And this will lead to EPA with a 20 carbon. And then you have several steps. You got elongation, you get desaturation, it involves peroxisomes, and then you finally get docosahexaenoic acid. And it's going to be very high levels in the brain. Now, the omega-6 precursor, linoleic acid, is going to again go through a very slow desaturase process, and you're going to eventually get to arachidonic acid. And so this is going to go through the ER. You have a delta 5 desaturase involved, very different enzymes, and eventually you're going to get your arachidonic acid. So arachidonic acid is going to come from your omega-6 fatty acid. Now, some differences between your omega-6 versus your omega-3 is that you're going to have a difference in their actual pro-inflammatory or anti-inflammatory functions. So with omega-6, you're going to mostly have incorporation into membranes. And remember, I told you that your arachidonic acid usually incorporates into that number 2 position of your glycerol backbone in your fatty acids in your membrane. They are going to form arachidonic acid, and they're typically going to be pro-inflammatory. Your omega-3 will also incorporate, so that's the same thing, it can incorporate in the membranes. It's going to form precursors for different molecules called resolvins and protectins. So they're going to primarily be anti-inflammatory. You have a D series, and this comes from DHA, and then you have your E series, and this is from our EPA. Note that DHA is also important for normal brain development. Okay, so we have our resolvins. Resolvins are going to be a modulating lipid coming from that omega-3 fatty acid. And again, we have our E series from EPA. And these are going to be increased following aspirin or consuming EPA. They are going to reduce inflammation. They are going to reduce immune cell function, specifically at a cell population called dendritic cells. And dendritic cells are cells of the immune system that actually get your T cells activated. And they can also help to resolve inflammatory responses. Now your D series from DHA, this is going to again be formed in the presence of aspirin and DHA. Sorry, I was going to sneeze. Okay. These guys are actually going to, instead of block the activation of T cells, they actually block T cells in their function. They're also going to block migration of your polymorpho nuclear cells. And your polys are cells like your neutrophils, your basophils, your eosinophils. So those are the PMNs. They're going to promote T cell apoptosis. They're going to decrease inflammatory cytokines. And inflammatory cytokines are the communication of your immune system. So it's a way, they use cytokines as a way to tell other cells to get activated, and so if you decrease those, you decrease activation. And they can reduce airway inflammation. Okay, so one of the first things, of course, that has to happen is that you have to make these different compounds get arachidonic acid out of the membrane, or whichever fat you're working off of. So we talked already about lipoxygenase. Or phospholipases. Oxygenase will come in a minute. We talked about our phospholipases. We have phospholipase A1, A2, that would remove either the fatty acid 1 or the fatty acid 2 from a glycerol backbone, as shown here. We also have phospholipase 3, which is able to remove from that 3 carbon. And in this case, it's just showing a phosphate group, but you can imagine that that could also be potentially another fatty acid present there, which sometimes it is. doesn't always have to be a phosphate group on that position 3. And then your phospholipase D will remove whatever that head group is, whether it's a serine, ethylamine, or whatever is in that position. So the first thing you have to do is to get out your arachidonic acid. And arachidonic acid is usually at this position 1, and so that would require phospholipase A2. So your phospholipase A2 can remove arachidonic acid directly from your 2 position of your fatty acid. Now, there is another way to get arachidonic out of your lipid, and that is through phospholipase C. So phospholipase C is going to remove whatever's attached to that 3 carbon, and in some cases that's going to be phosphatidylinositol bisphosphate that it's going to work off of in your membrane. And then when this happens, you're going to release diacylglycerol, which you already know, because from phosphatidylinositol bisphosphate you can get diacylglycerol or DAG. And in the cell you have diacylglycerol lipase, and this can generate arachidonic acid. It'll also generate monoacylglycerol, and then this can generate through a monoacylglycerol lipase arachidonic acid as well. So you in essence have a few ways, your cells have a few ways to tap into different fats in the lipid membrane, whether it's a TAG or a phospholipid, or whether it's specifically phosphatidylinositol, and you can break those down to get at your arachidonic acid. So with respect to icosanoid synthesis, there are going to be 2 main pathways, but some things to note. All cells except red blood cells can synthesize icosanoids. So they all can do this. Icosanoids are extremely potent, so a little goes a long way. So you don't need much. And all icosanoids work locally. So they're going to be autocrine or paracrine. And so that's why it's important that all cells are able to make them, because you don't know where in the body you're going to need thromboxin, for example, or other types of icosanoids. Now again, the 2 main pathways are going to be a cyclic pathway and a linear pathway, and the trigger is always going to be G protein receptors, so it's going to be something triggering a G protein. So we'll study this a lot when we study the platelets. We'll talk a lot about the activation of platelets with icosanoids, as well as inactivation of platelets. So again, some icosanoids counter each other, and their balance is actually really important for homeostasis. All right, so in the cyclic pathway, you have some stimulus that is going to trigger that a G protein. We have epinephrine, we have thrombin, and bradykinin listed here. And the first step is going to be to activate your phospholipase A2. And so you can imagine that you have a cell membrane. And on the cell membrane, you're going to have your G protein receptor. And so you're going to get some signal. Right? And then your signal is you're going to have a G alpha receptor. And so you have a G alpha activating as well as a G alpha potentially inhibiting. So your G alpha and we'll put a G alpha. Oops. And so our G alpha can open a calcium channel. And let calcium in. Okay, and then our G-alpha can also activate let's do a different color. So we can get phospholipase C, and then we have PIP. Phospholipase C can interact with PIP, and you get DAG plus IP3. And then you also have IP3 that can go into the endoplasmic reticulum. So here's our ER. And we have a calcium channel in our ER. So here's calcium. And in this case, calcium is going to come out this way. And then our protein kinase C can interact with your fats in the membrane of your ER. So in the membrane of your ER, you can have your phospholipases, and so you're going to have your protein kinase C and protein kinase C can phosphorylate your phospholipase A2. And this will activate this, and it will release arachidonic acid. Let's make this from the membrane. Now, calcium that also is coming in can also activate protein kinase C, and it can also activate your lipase A2. So there's many mechanisms through signaling and different G protein receptors that you can get release of arachidonic acid from your membranes. Okay, so what happens? Oops, sorry. So what happens with arachidonic acid? Okay, so you have arachidonic acid released from your membranes, and now it's going to have to start making things. So you have some enzymes, cyclooxygenase, which is going to form prostaglandin G2, which is shown here. Then prostaglandin G2 is going to be metabolized by a peroxidase, and this involves our glutathione. And then prostaglandin G2 will produce prostaglandin H2. And prostaglandin H2 is sort of like the, if you think of a train station, it's sort of like the place where you can go in multiple directions. So from prostaglandin H2, you can generate prostaglandin D2, which is a precursor for F2 alpha. I'm sorry, I'm like way ahead of myself here. Prostaglandin E, so prostaglandin H2 can become prostaglandin E2, and E2 is the precursor for F, not the H. So you get F2 alpha. And then, of course, H2 you can get prostaglandin GI2 or thromboxin A2. So what's important to note about these two is these two often are opposite of each other. So when you have thromboxin activated, oftentimes to counter it, you will get prostacyclin or prostaglandin I2 activated. So one's going to cause vasodilation, one's going to cause vasoconstriction, one's going to activate platelets, one's going to inhibit platelets, etc. So those two in balance are extremely important. They're antagonistic of each other. Okay, so let's just go through each of these just a little bit. We'll talk about them. H2 is generally just a precursor. So that's really what you have to remember about H2. It doesn't really have a function other than to make something else. prostaglandin I2 or prostacyclin is again going to be important for countering thromboxin, especially during clotting. So it's going to be a vasodilator. Its common name is prostacyclin. You might have heard of it that way. And importantly, it's going to prevent platelet activation. So when we talk about platelets, you're going to see prostaglandin I2 and thromboxin interact, and they are going to be made during the coagulation process, especially during platelet activation. Prostaglandin D2 is important for your mast cells. Your mast cells are, of course, known for the allergic response, but they're also important for parasitic infections. So mast cells. And so they're going to have vasodilation, bronchial constriction, hard to breathe, which many people who suffer from severe allergies know. Prostaglandin E2, which is referred to as dinoprostone, this is going to have an abortifacient reaction. So this is going to have effects on uterine contraction and cervical ripening. It also has other effects. It is going to promote inflammation. This is prostaglandin, so it promotes inflammation. It has a bronchoconstriction as well as a vasodilation, so has a similar effect as your prostaglandin D2. And then you can also have some increase in body temperature associated with E2. The prostaglandin F2 alpha is dinoprost, and this is also an abortifacient. It also has an impact on uterine contraction, similar to our prostaglandin E2, and it is a bronchoconstriction, which is similar as well. And just keep in mind that F2 is made from E2, and it's simply just a reductase that changes E2 into F2 alpha. And then we have thromboxin A2. Thromboxin is, of course, going to be produced by activated platelets. It has a very, very short half-life. It's going to be involved in platelet aggregation, and it has a cascade. And of course, your thromboxin I2 or your prostacyclin is an antagonist for thromboxin A2. When we talk about your clotting cascade, this is what this means. It works in a cascade, so your thromboxin is part of this whole clotting cascade. Now, thromboxin B2 is very important because A2 has seconds of a half-life. That is simply because of B2. B2 is a product of the A2 breakdown, and which happens very quickly, and it's through a thromboxin. It's almost like it happens spontaneously. So A2 will break down into B2. So B2 is not functional. It does not have a function except to be a byproduct of A2. So your prostaglandins, your thromboxins, are going to have to be regulated. They are going to be inactivated, and inactivation can occur from seconds to minutes. Oops. They can be inactivated. They can be oxidized by a 15-hydroxyl group, which is critical for their activity. So if you oxidize this 15-hydroxyl group, then you get a ketone and they become inactivated. You can also have reduction of a double bond at position 13. And you also have beta and omega oxidation, and this occurs at the portions of the non-ring. And this is going to produce dicarboxylic acids, and all of these are then just going to be simply secreted, and they get secreted in the urine. You also have your thromboxins. So your thromboxin A2 rapidly becomes thromboxin B2. And this is going to be due to a cleavage of that oxygen bridge. And so A2 is the active form, and B2 is inactive. And it'll just be secreted. All right, so now there's leukotrienes, and leukotrienes are going to be made through what's called a linear pathway. And this is going to be through arachidonic oxygenases, or your LOXes. So you have three main LOXes, 5 LOX, 12 LOX, and 15 LOX. They're all enzymes like oxygenases. There's just simply called arachidonic 5 oxygenase, or 12 or 15, for example. The 12 and 15 can also be involved in the synthesis of our lipoxins, but your 5 cannot. Your 5 has nothing to do with lipoxins. So your formation of your leukotrienes is going to involve your removal of arachidonic acid, similar to the icosanoids. And then you will get arachidonic acid, but instead of cyclic oxygenase, you get lipoxygenase that works off of it. In this case, we're showing 5 lipoxygenase. And so for 5 lipoxygenase, you are going to get the intermediate of your 5 hep8. And then this is going to form leukotriene A4. And leukotriene A4 can become leukotriene B4, or it could become C4. And then C4 can, through various steps, including your removal of glutamate to form a D4, and then your removal of glycine to form an E4. So basically, lipoxygenases are going to incorporate oxygens into the carbon of several double bonds of arachidonic acid. And this is going to form a hydroperoxy group. At these points, the double bonds are going to isomerase, and one carbon is going to be removed and transferred from cis to trans. And this unstable hyperoxy group is converted to a hydroxy group. Now, the functions of leukotriene B4 is going to be to increase vascular permeability, it's going to increase T cell proliferation, it's going to increase your leukocytes, white blood cells, aggregation. It's going to increase what's called interferon gamma production and increase pro-inflammatory IL-1, interleukin 1, and IL-2. So IL equals interleukin. And so those are communicators of the immune system. So all of this all together should trigger, it's going to be pro-inflammatory. Now your leukotriene C4 and D4 will also be bronchoconstrictive. They are going to increase vascular permeability. and increased interferon gamma. And so they are also going to be pro-inflammatory. Okay, I had to let one of my dogs out. Sorry about that. Okay, so then, as I said before, your lipoxins can also be formed from your 12 arachidonate, 12 lipoxygenase, or your 15 lipoxygenase. And so you're going to have 3 different pathways of producing these lipoxygens. And they're going to be what's referred to as a classic pathway, which is going to be in our white blood cells, our leukocytes. So these are our white blood cells. You're going to also have an epithelial pathway, epithelial cells, and as well as aspirin can initiate the production of lipoxins. So in the classic pathway, you basically have a leukocyte that's going to interact with a platelet. Okay, so we have our leukocyte, our white blood cell, and I'm going to make a platelet very large so I can write in it, but platelets are usually much smaller than this. And so you're going to have a G protein receptor on the surface of your leukocytes. And so you have some sort of stimulation, and then this is going to stimulate arachidonic acid being removed from your lipids. So we'll put PIP2 here. And so you're going to get your G protein activated. You're going to get your lipoxygenase A2 activated, and then you're going to release arachidonic acid. Now, arachidonic acid is going to undergo your lipoxygenase pathway. And so this is going to be a 5-LOX pathway. And you're going to get leukotriene A4 made. And then leukotriene A4 can be hydrolyzed to leukotriene B4. And then this is going to be pro-inflammatory, as we just talked about. Now, leukotriene A4 can also be transferred to your platelet. And leukotriene A4 in your platelet will be worked on by a 12 lipoxygenase. It could also be 15, but by a 12 lipoxygenase. And this can form lipoxygen A4 or lipoxygen B4. And these are just going to have different enzymes that work on them. So you're going to have a lipoxin hydrolase or a lipoxygen B4 hydrolase. And then both of these are anti-inflammatory. And so you can imagine that the immune system does this on purpose to balance. So first you're going to get a pro-inflammatory, but you want to make sure that you don't cause too much inflammation because you're going to damage tissues. And so you can also, at the same time with the same intermediate, generate anti-inflammatory products as well. So the next pathway is an epithelial pathway, which is going to involve that 15 LOX as well. And this is going to have the flips. So you have a 15 lipoxygenase in epithelial cells that is then going to have an intermediate transfer back to your leukocytes, and that's going to be worked off by a 5-LOX pathway. And then finally you have your action of aspirin. So aspirin is going to inhibit cyclogen oxygenase 1 and 2, and it's important to understand what the difference between cyclogen oxygenase 1 and cyclogen oxygenase 2 is. COX-1 is going to be constitutively expressed, which means it's always on. 2 is inducible. So 2 is going to be inducible in immune cells only, our white blood cells. No other cells in our body are able to do cyclogen oxygenase 2. It is going to have the same exact function as cyclogen oxygenase 1. It's just that during inflammation, you're going to double the amount of your icosanoids produced, because now you don't only have just cyclogen oxidase 1 working, but you also have cyclogen oxidase 2. So in endothelial cells and epithelial cells, acetylation of cyclogen oxidase 2 is going to alter the enzyme, and this is going to form a 15R hydroxy icosanoid icosanoids tetragenic acid. Okay, so what's going on here? So with respect to aspirin, you have again your endothelial cells and epithelial cells. Okay, so here's our endothelial cell, epithelial cell. And then you have over here your white blood cell. So we'll do a macrophage-like thingy. Here's our white blood cell, or a neutrophil, or something like that. And so in your endothelial cell, epithelial cell, you'll have aspirin come in. Okay, and then aspirin will interact with cyclogen oxygenase 1. Okay, and so cyclogen oxidase 1 is going to form in the epithelial cell our 15R het. And our 15R het is going to go into your polymorphonuclear cell. Ooh, uh-oh, I don't know how I did that. Okay, good. Okay, it's going to go into our polymorphonuclear cell. And it's going to be worked on by our 5-LOX system. And it's going to generate our A4 and our B4, which is going to be anti-inflammatory. Okay, so you're basically causing your immune cells to generate anti-inflammatory icosanoids by having aspirin present, which changes cyclogen oxygenase activity. So this is going to block the prostaglandins and push more towards your lipoxins, which are more anti-inflammatory. Okay, so just really quickly, there are icosanoids receptors. They're all going to be G protein receptors. We have 10 prostaglandin receptors, and they're going to be D prostaglandin, E prostaglandin, F prostaglandin. Ds are going to bind to the D families, E's, F's, etc. We also have prostacyclin receptors, and those are going to be the IP receptors. Thromboxin receptors are the thromboxin are going to bind to thromboxin A2, not B2. And then we also have 4 leukotriene receptors. We have our leukotriene B4 that are BLT1 and BLT2. We also have our peptidyl leukotriene receptors, cysteine LT1 and cysteine LT2. So icosanoids are typically pro-inflammatory. Change this color. Okay, so we have our series 2. And these are going to be our prostaglandins and our thromboxins, and these are going to be pro-inflammatory. So those are pro-inflammatory. And then we have a series 4 leukotriene. And this is very pro-inflammatory. So there are non-steroidal anti-inflammatory drugs, or NSAIDs. And these are going to target both cyclogen oxygenase 1 and cyclogen oxygenase 2. And then we have, these are going to be aspirin, ibuprofen, naproxen, phenylbutazone are all going to be examples of those. There are some cyclogen oxygenase 2 specific inhibitors as well. So those will just be more used more during an actual inflammatory process. So cyclogen oxygenase, again, 2 is only going to be during inflammation. So this is constitutive. And this is only white blood cells during inflammation. Your NSAID drugs target both. You have drugs that target cyclogen oxygenase, and these are going to be like Cerebrix, Vioxx, and those are going to not be specific for your cyclogen oxygenase 1. So this will be Celebrex, no. Okay, so using your non-steroid or anti-inflammatory drugs to treat inflammation has led to the development of ulcers. And so that's why the development of our cyclogen oxygenase 2 only drugs has been so important. There are some cardiac benefits of aspirin, but these benefits are neutralized if you're taking them with a cyclogen oxygenase 2 specific inhibitor. So our cardiac All right, so just a side note about our corticosteroid drugs. These are going to target your phospholipase A2. And so they're going to prevent or slow, I should say slow, because we have our lipase C that can also tap into and get arachidonic acid out. But it's going to slow down the release of arachidonic acid from the membrane, and so that's going to, of course, slow down the production of your icosanoids. So here's an overview pathway of all the arachidonic acid metabolites or active lipids that we've been talking about. So we have our cyclogen oxygenase pathway, our lipoxygenase pathway. You're going to get your prostaglandins, thromboxins, pro-inflammatory. You're going to get some leukotrienes, pro-inflammatory. Lipoxygens are going to be anti-inflammatory. And we also have another series called epoxides. And our epoxides are going to involve a cytochrome P450 pathway where they use arachidonic acid to generate your epoxides. So cytochrome P450 we've already talked about. These are going to be in the endoplasmic reticulum. And this is going to oxygenate arachidonic acids. And of course, whenever you do this, you have the potential for active oxygen species. This activity is of the monooxygenases in the microsomal system. So we have our and this is basically our P450s, and these are going to yield our epoxides. And then you can also form different dihydroxide or dihets can also be formed. Biologic activities of these compounds are going to be, they're going to have activities in your ocular, eyes, vascular, endocrine, and renal systems. So let's just talk a little bit about how icosanoids actually function. What is their action? Now you have icosanoid receptors, and icosanoid receptors are going to either have activating or inhibiting functions. So they are G protein receptors. So just like all G protein receptors, they can activate the adenylate cyclic AMP. And then you get your pKa, right? And the ones that do this are going to be your prostaglandins E, D, and I series. And then you can also have that they increase calcium. And so this is going to be your prostaglandin F2A. Or alpha. Your thromboxin, remember only A2. Leukotrienes are also going to do this via the calcium. Icosanoids can also bind to subunits of the G proteins within the plasma membrane, the target cell. If it binds to a stimulatory G protein, I'm just going to write it as GS for stimulatory. Then you get amplification. If it's going to bind to inhibitory, then you're going to get a reduction. In the cellular response. So this is the cell response. Okay. Now, isoprostanes are going to be a byproduct of arachidonic metabolism, and it happens by lipid peroxidation initiated by free radicals. So this is not the cell trying to tap into arachidonic acid because it needs it to make icosanoids. These are free radicals doing damage to the lipids, and it's going to release arachidonic acid. Now, arachidonic acid, when it's part of that phospholipid at position 2, can undergo free radical damage. And then what happens is phospholipase A2 will remove the altered icosanoid and try to replace it with a good icosanoid. And this is going to release it into the circulation. So damaged arachidonic acid is actually a useful biomarker for patients that have a lot of oxidative stress, have a lot of lipid peroxidation occurring. So just keep that in mind. So during oxidative stress, you have damaged arachidonic acid, and it's going to be removed by that phospholipase A2 and released. Oops. So therefore, an increase of damaged arachidonic acid indicates oxidative stress. Okay, and so here is your arachidonic acid, again in your R2 position. You have your radicals do damage, so now you have a R2 that has a star because it's been damaged. Phospholipase A2 will remove that, and then you get the damaged arachidonic acid, which is called isoprostate, circulating. So increase of isoprostate is going to be an indicator of stress. Also have endocannabinoid synthesis. These are endogenous ligands, so something that can bind to the cannabinoid receptor, and they're going to be primarily effective in the nervous system. So endocannabinoids are ligands. An anandamide is going to be synthesized in neurons, and this is going to be starting with phosphatidylethylamine. And this occurs in neurons. Intracellular calcium elevation is going to be what's important for endocannabinoid synthesis, and calcium is going to increase due to depolarization following a neurotransmitter on the postsynaptic neuron. So you have neurotransmission. Spelling that wrong, sorry. So that's going to lead to the increase of calcium. Now the calcium influx is going to activate a transacylase. And that's TAE. And then so your phospholipase D, remember phospholipase D is going to cleave whatever's attached to this phosphate group here, right? So you're going to have phospholipase D is going to cleave your NAP, which is this product, into an anandamide here. So again, we have TAE, which came from an increase in calcium source, and then that is going to generate your NAP, and then that is going to eventually be cleaved to yield the anandamide. So this whole piece right here is going to be cleaved by your phospholipase D, and you're going to generate that anandamide. And then once that is released, it's going to be a retrograde messenger, and it binds to receptors on the presynaptic membrane. And its binding is going to alter the ion influxes such that you have neurotransmitter release from the presynaptic neuron can be increased, and it results in an analgesic effect. Okay, so we will stop here for part 1.